Question, would an anabolic reaction be endergonic or exergonic, and why? Well, the board kind of already tells you the answer to that. Remember, anabolic is putting things together. Does putting things together require energy input, or does it release energy? Putting things together requires energy input, and that would be endergonic. Endergonic. Putting things together. Energy input required. Reactants have less energy than the products. Okay, now we need to talk about a very specific type of energy called activation energy. Because even if a reaction releases energy, it doesn't usually start spontaneously. It makes sense that this wouldn't start spontaneously, right? You go to IKEA and you buy the dresser in the box, and you don't just leave it on your floor and expect that it's going to assemble itself, unfortunately. So it makes sense that this would require some energy to start it. But exergonic reactions actually require energy to start also. If you think about it, that boulder at the top of the hill is just sitting there. It's not going to roll down the hill until it gets a little push. And sometimes that push is a lot. Sometimes the push required to get that ball rolling down the hill in this downhill chemical reaction from an energy standpoint, sometimes that push is greater than the energy that's available. That push that's required to get a reaction started is called the activation energy. Also sometimes called the energy of activation, EA. So activation energy. You'll often see a capital E with a superscript A. Okay, this is the energy required to start a chemical reaction, even if it's exergonic. So on the screen is a little more scientific definition. Energy required to destabilize existing bonds and initiate a chemical reaction. So even when we're breaking bonds, exergonic, we're going to need energy to break those bonds. Remember, I already mentioned that covalent bonds are hard to break. When they do break, they release energy, but we're going to have to do something to those bonds to get that breaking started. Enzymes play a very important role in this. What do enzymes do? Enzymes lower the amount of activation energy required to start a chemical reaction. They are called catalysts. So catalysts lower the amount of EA required to start a reaction. Lower the amount of activation energy required to start a reaction. And the chief catalysts are enzymes. Enzymes lower the activation energy required to start a chemical reaction. And here's a very good example of that. This shows the amount of free energy supplied to get this to go from reactant to product. If it was just going downhill without this input of energy needed, it would just go downhill. But you see, we're going to have some energy input. We're going to have to push that boulder before it starts going down the hill. Okay, this is the amount needed when there's a catalyst present, chiefly the role of enzymes. So catalysts lower the amount of activation energy required to start a chemical reaction. That is the exact same slide. Sorry about that. Okay, more terms. We're going to get back to enzymes in a minute. There are just a couple of more terms you need before we talk more specifically about enzymes. Boy, oh boy, more terms, more terms, more terms. So much vocabulary in a general biology course. Almost like taking a foreign language. There's so much. Redox reactions, two important terms here. Oxidation and reduction. These two always happen together. One cannot happen without the other. I like to think of it really as the transfer of electrons, which we've already looked at when we looked at ions forming and then ionic bonds. But we're going to put some different terms on this now, because sometimes we're not just forming ions. Sometimes we're doing something considerably more important. Okay, so oxidation and reduction always go together. And when they're together, we call that a redox reaction. I'm going to give you a couple of definitions, and then I'm going to give you an example. So oxidation, this is defined as the loss of electrons. They're not just lost to the universe, though. They're actually transferred to another atom or molecule. So reduction is the gain of electrons. Loss of electrons, gain of electrons. Where were those gained from? They were gained from whoever lost them. That's why the two always go together. And we call that redox, oxidation-reduction reaction. The way to remember which is which sounds really silly, but I promise this will serve you well if you remember this. Loss of electrons is oxidation. Gain of electrons is reduction. Leo the lion says grrr. I know that sounds silly, but it works. Leo, loss of electrons is oxidation. Gain of electrons is reduction. Why do we care about this? This is going to be a very, very important part of the energy transfer when we're going through the steps of cell respiration. It's also important in photosynthesis. So redox reactions are important to understand. Loss of electrons, gain of electrons. I'm going to go back to the example we used with sodium and chlorine so you can relate it to something you already know. Remember, sodium is atomic number 11 and chlorine is atomic number 17. If we just look at electrons in the outer shell, remember that means sodium has 1 in the outer shell and chlorine has 7. When chlorine transfers 1 electron to chlorine, sodium now has 10 and chlorine has 18. Sodium lost an electron. Loss of electron is oxidation. We would say that sodium was oxidized. It lost an electron. Did it lose an electron to the universe? No, it lost that electron to chlorine. Chlorine gained an electron. Gain of electron is reduction, grr. So we would say that chlorine is reduced. So oxidized, reduced. This is oxidation, and this is reduction. The slide I'm going to show you is just a really general example. The one I just gave you is more specific. Okay, so if that electron is transferred from A to B, we produce ions, yes. A lost an electron. That would be oxidation. So we would say that A is oxidized and B is reduced. ATP. Again, we're just going through a lot of definitions here. We're going to put this all together in one big story when we talk about cell respiration. ATP, adenosine triphosphate. This is the energy currency of all cells. You already know that. We talked about that before. This is what it looks like. In its most simple cartoon form that we're mostly going to draw, here's our adenosine part of the molecule, and then triphosphate. 1, 2, we draw a squiggly line for that last high energy bond. It's a covalent bond, but we're going to call it a high energy bond, and that will get explained later. So this would be triphosphate. We also have ADP. The adenosine plus 2 phosphates would be ADP. And we also have AMP, adenosine monophosphate. And that's what this picture is showing in a little more detail. Okay, so this would be AMP. What does this look like to you? A pentose sugar, a base, and a phosphate group. What other macromolecule has that as a monomer? It looks just like a nucleic acid. It looks very similar to RNA. It has ribose, phosphate group, and a base. Now we have another phosphate group and another phosphate group, 3 phosphate groups. When we say that we're making ATP during cell respiration, we're not making ATP. We're not taking all of these atoms and putting them together from scratch. When we say we're making ATP, all we're doing is we're taking ADP and we're adding a phosphate group back on. Transfer of this phosphate group to another molecule equals energy transfer. When that phosphate group gets transferred, it's called phosphorylation. This is a very important term. Phosphorylation. Phosphorylation is transfer of a phosphate group from one molecule to another. It's a very general term, but it applies in this case. ATP is going to transfer this phosphate group to another molecule, and when it does that, it will become ADP. So phosphorylation, transfer of a phosphate group from one molecule to another. So I want to draw the specific example I'm speaking of, ATP. This is a very important part of the story. This is the whole point of cell respiration, is to regenerate ATP after it's used. So here's our adenosine triphosphate. Squiggly bond, high energy bond. We're going to take this phosphate and we're going to transfer it to another molecule. I'll make that molecule square. So here's the other molecule. Transfer of this is going to lead to that molecule now having a phosphate group attached. It's now energized. But now guess what? ATP lost a phosphate group. Now it's going to be ADP. It's adenosine diphosphate. ADP. This transfer of a phosphate group is called phosphorylation. What do we have to do to regenerate ATP? We need to stick a phosphate group back on. We need to add this back on. We need to rephosphorylate ADP to form ATP. That's what we're going to do during cell respiration. So we're never making ATP from scratch. We're rephosphorylating ADP. How do we get ATP? Because we transferred this phosphate group to another molecule to cause a chemical reaction to take place. It equals energy transfer. So ATP hydrolysis drives reactions, specifically endergonic reactions. Endergonic reactions require energy to take place. Okay, it's Reactants lower energy than the products. Energy input is required. Breaking this bond and transferring that phosphate group to another molecule, that's going to drive that reaction. ATP is an energy currency, but I just want to note it is not a long-term energy storage form. We use it within a few seconds. I've heard that we use our entire body weight of ATP in one day. We're just constantly making it and using it. That's not how we store energy. We store energy first as carbs, later as fats. Okay, and finally a little bit more information about enzymes. A little bit more detail about enzymes. There are lots of enzymes required to carry out cell respiration. Lots of enzymes required to carry out cell respiration. So enzymes are required even for exergonic reactions, not just endergonic reactions. So enzymes, a little bit more detail. Remember that most enzymes are proteins. In fact, most of our proteins are enzymes. Most of our DNA that codes for proteins is coding for enzymes. We do have some enzymes that we've discovered that are RNA enzymes, but we're only going to focus on the protein enzymes in this course. These are the catalysts. They lower the activation energy required to carry out a chemical reaction, whether it's endergonic or exergonic. It's important to note the enzyme is never used up in a chemical reaction. It gets recycled. It gets reused over and over. It's not just one shot. But it is very reaction specific because one enzyme only binds one substrate. Enzymes are very substrate specific. And that's because of this lock and key. The substrate has to be able to fit into the active site of the enzyme. Shape is key. We talked about this a little bit when we talked about proteins, that if we do anything to alter the shape of that enzyme, that substrate might not fit anymore, and that chemical reaction won't take place. So, remember the substrate, that's the reactant. Okay, and the substrate binds to the region called the active site of the enzyme. So this is showing a substrate coming into the active site of an enzyme, and it has to fit correctly. If it doesn't fit, the reaction doesn't take place. Shape is key. This is a little more information than you really need to know, but what's important to realize is that that enzyme, so let's use sucrase. We've used sucrase before as our example. I can't remember the specific shape I gave the active site then. Remember, sucrase is the enzyme that breaks down sucrose. ASE tells us that it's an enzyme, and enzyme names tell you what they do. Sucrase is going to break down sucrose. Remember, sucrose is fructose plus glucose. Okay, so I'm going to make my pretend sucrose molecule look like this. Fructose, glucose. This is our sucrose. This is going to be the active site of that sucrase enzyme. Okay, in sucrose, that's the substrate. This is very specific, this relationship. Lactose would not fit into the sucrase enzyme. Lactase is going to have a different shape. It's going to be glucose plus galactose. It will not fit into that active site. This is a very specific relationship. What's going to happen is that sucrose molecule comes in and fits into the sucrase, and that enzyme through something called induced fit, is going to put pressure on this bond. And now that bond is going to break with less energy required. It's kind of like, rather than just two puzzle pieces fitting together loosely like this, it's actually squeezing on the substrate. It's putting pressure on that bond. Induced fit. It's a very tight fit. Pressure's put on that bond, and now those products are released. Enzyme gets used again. Keeps getting used over and over and over. The enzyme is not used up, it's not consumed in the reaction. There are a couple things that are going to affect enzyme activity. One is going to be concentration of the substrate in the enzyme. If you take the lab for Biology 102, you will do an enzyme experiment. Same for Bio 100, you'll do an enzyme experiment. And the concentration of that substrate is going to be a factor, as well as the concentration of the enzyme that's present. Also, we've already talked about the fact that changes in temperature and pH can dramatically affect enzymes. Because if we denature this enzyme and change the shape of this active site, that enzyme is not going to function properly. So it's important for the body to maintain constant pH and constant temperature. Normal for whatever that environment is. This shows you that really pH and temperature, what's normal depends on what's normal. I guess is the simplest way to put that. Normal isn't always 98.6 degrees. Normal isn't always pH 7. So it's whatever is normal for that enzyme, where it normally exists in the body, whatever the normal environment is. That's what we're trying to maintain. And remember, we use buffers to maintain that pH. Sometimes we want to inhibit enzyme activity. What if we didn't want sucrose breaking apart right now? So long as the sucrase is present, it's going to happen, unless we do something to stop it. And that's what enzyme inhibitors do. Enzyme inhibitors are how we control enzyme-activated reactions. So these control enzyme-catalyzed reactions. We're going to see a very important example of this when we talk about cell division. We don't want the chromatids pulling apart until we've done this checkpoint to make sure everything is all connected correctly. There are these protein complexes called cohesins holding that chromosome together, the two sister chromatids. We don't want that enzyme going in and digesting those cohesin proteins until we're ready, so we use an enzyme inhibitor. Enzyme inhibitors are very important. That's how we're going to control the reaction from taking place. Two ways you can do this. You can either use an inhibitor that directly blocks the active site. So if I had an inhibitor in here that directly blocks this active site and keeps sucrose from binding to the sucrase enzyme, this would be called a competitive enzyme inhibitor. It directly blocks the active site. So you can see that here. Competitive inhibitor competes with the substrate for the active site. It actively blocks the active site. There's another category called a non-competitive inhibitor. And what that does is it binds somewhere else on the enzyme, not in the active site. But when it's bound, it changes the shape of the active site so the substrate can't bind so long as this inhibitor is present. As soon as the inhibitor is released, the reaction can take place. So this would be a non-competitive enzyme inhibitor. Again, it binds somewhere else on the enzyme, but it changes the shape of the active site by binding. So it causes shape change that makes the enzyme unable to bind the substrate. Okay, so this would be a competitive inhibitor right there in the active site. This would be non-competitive, and you can see when that's bound, it's going to change the shape just enough that that substrate can't bind to the active site. And that's the end of this energy lecture. I know it's a lot of terms. If there were any terms that were unclear, please rewind. We're going to use all of these terms in understanding the story of cell respiration, how we extract energy from our food molecules, and use that to drive chemical reactions in the body via ATP.